So again, if you don't already know me, I'm Alexa Tinsley. I'm the Director of Programs here at Jett Foundation. I want to thank you all so much for joining us today on our webinar series. Today, we are welcoming REGENXBIO to hear about their RGX-202 Affinity Duchenne pivotal trial and interim functional data. We are so grateful to our partners like REGENXBIO for making these webinars possible and bringing all of these right into your home virtually. Today, we welcome Dr. Naz, I'm told her I was going to get it wrong, Dasgir, Pediatric Neuromuscular Specialist and Clinical Development Lead of the RGX-202 program at REGENXBIO. So before we get started, I do just want to mention a few things. This is being recorded and also live streamed to Facebook. So if for any reason you have to hop off, or if there was anybody that you know that wanted to attend today's webinar but wasn't able to, you will be able to share it. It will be on our YouTube after we get the recording, and it will also be live and ready and recorded on Facebook right after the event. There's also closed captioning. So if you need any closed captioning, at the bottom of your screen, there is a little box that says CC, so show captions, and you should be able to translate into any language of your choosing. We will also have a live Q&A at the end. If there are any questions that you have, we do ask that they go in the Q&A box, which is also at the bottom of your screen. There's a little box with a question mark in it. We do primarily use those to answer questions. We do try to monitor the chat, but we do primarily answer questions through that Q&A panel. For anybody watching live on Facebook, we do also try to monitor those questions and try to ask them in this presentation as well. But for any questions that we don't get to, or if for some reason you aren't here and watching the recording, please do feel free to reach out to us and we will try to get those questions to the REGENXBIO team after the fact as well. So that is all from me, and I will hand it off to Naz.
Thanks, Alexa. All right, thanks everyone for your attention today. So as Alexa mentioned, we're going to be talking about the Affinity Duchenne study, which is now in its pivotal phase. So we're out of the phase 1/2 portion of the study. And then we're going to be talking about some interim functional data that we've gathered so far from our phase 1/2 portion of the study, which had the primary goal of safety, but we obviously got some functional data as well. I have to just provide these forward-looking statements about our company. All right, so I'll tell you a little bit about REGENXBIO. You may be familiar with us, maybe not, and talk about our drug RGX-202. Our study for Duchenne muscular dystrophy gene therapy is known as Affinity Duchenne. So we'll talk about the phase 1/2 study, give you an overview, review the data, and we'll then go into what our phase 3 study is designed as. And then finally, we'll have a question and answer session, and no question is inappropriate. So please feel free to ask, even if it's not necessarily directly related to our study, but just questions in general about gene therapy. And if there's something I can answer, I'd be happy to do that. All right, so REGENXBIO, as a company, we are seeking to improve lives through the curative potential of gene therapy. We pioneered the landscape of AAV, or adeno-associated virus, gene therapies. As you know, adeno-associated viruses are typically used for gene therapy. They're one of the vehicles used for gene therapy. And these are just little viruses that were found on the plate growing with adenovirus, a different type of virus. And thousands of patients have been treated with this approved and with approved and investigational medicines built on our NAV technology platform that utilizes AAV gene therapy. We are advancing late-stage gene therapies for patients with retinal and rare diseases. Our lead candidate is in Duchenne muscular dystrophy. And we have expertise with end-to-end capabilities. So we not only do research and development and then clinical development, we also have fully integrated manufacturing and fill-finish capabilities, which means bottling the drug, whatnot, making sure it's good to go and be used clinically. So we have the capabilities to support multiple potential product launches. So let me talk a little bit about RGX-202. So RGX-202 is the name of our drug that we're using. It's our gene therapy to treat Duchenne. It is known as a microdystrophin gene therapy, and it is the only microdystrophin gene therapy with the functional elements of the C-terminal domain. The C-terminal domain is found in naturally occurring dystrophin. So let me kind of go through some of the different drugs that are out there and how we compare to them. So, as I mentioned, there are adeno-associated viruses. Adeno-associated viruses may be of different types, and they're basically vehicles for the delivery of the drug. Our AAV is known as AAV8. There is Sarepta's drug that uses AAV RH74. Solid Biosciences uses SLB101, and Genethon also uses AAV8. Then you have a promoter which kind of helps get your transgene to the correct location. So we use SPC512, and obviously the different companies use different promoters below. The promoters are set in place to be specifically targeting the tissue that you're interested in treating, which in our case is muscle and heart. And depending on the size of the promoter, you can package as much as you can into the tiny vector. Unfortunately, these vectors aren't big enough to fit the entire dystrophin protein into them because it's very large. So they have to be kind of trimmed or tailored to the key elements of the dystrophin gene that you want to package into that. And that tailoring is variable between the different companies. And this includes ours, where we are actually able to include the C-terminal domain. So the C-terminal domain highlighted in green over here is where you can see that there's an ability to like differentiate us from the other companies and what they're able to package into their vector. So what is the role of this C-terminal domain? So let me kind of explain that to you too as well. So the C-terminal is some portion of it lives in the sarcolemma, which is the muscle in this it's attaches to the sarcolemma or the muscle membrane of muscle cells. And basically, we think that our C-terminal can bind there and kind of affix the microdystrophin protein and kind of anchor it in a good way to create stability to the muscle fiber itself, because there is some muscle fiber instability in the case of children with Duchenne or adults with Duchenne. So we're trying to kind of create, not only provide scaffolding to the location, but also make sure that that scaffolding has the right anchor to be in the right place. So basically, this is something that we developed based on preclinical studies or studies in mice that proved to us that, it's worth creating a drug that has the addition of this C-terminal domain because it may actually perform better, particularly in our human patients. So here you can see some of the data that we got from the mice. So here we see that the microdystrophin plus C-terminal domain better protects the muscle against contraction-induced damage. So as you know, contraction-induced damage is kind of like an eccentric contraction that can tear the muscle, and you can see that you can see some increased inflammatory cells or perfusion of the muscle tissue with something known as Evans blue dye that shows that there's problems with the muscle itself in terms of its susceptibility to damage with certain forces. So here you can see that there's information here that shows that you can create a good initial force, but there's also data that we've developed that shows that there's also less contraction-induced damage in the patients that have the C-terminal domain. So on the far left, you see the bar where there's the control mice and what force they can generate. So that's, just about at 70% initial force. Then we have the Duchenne mice, the DMD mice, also known as the MDX mouse, and you can see that kind of hits around 20% initial force. But when we provide our drug without the C-terminal domain to the MDX mice, that hits about 40%. And then when we provide it with the C-terminal domain, we get to around, just over 50% or so. So definitely seeing a benefit with the addition of the C-terminal domain. So this is on the next box here, you'll see how what we find has basically shown that the C-terminal domain may contribute to the improved outcomes with RGX-202. So the C-terminal domain significantly enhanced restoration of the dystrophin-associated protein complex in MDX mice. So you do have your dystrophin protein that attaches to this area, and the dystrophin-associated protein complex is just another group of proteins that lives in the muscle membrane. So we're hoping that that significantly enhances restoration of that and is more similar to dystrophin itself. then you also get prolonged microdystrophin activity. So the CT domain increased the half-life of RGX-202, which is how long it takes for 50% of the drug to exit your body, which remains in the muscle fibers longer to strengthen the muscle. And also the CT domain protected against contraction-induced damage, enabling better muscle recovery. So with that, we went ahead and got started with our phase 1/2 study, and here's an overview of the study and the data. So I'll go through this slide to kind of tell you what historically has happened, and things have changed a little bit with our pivotal study, so I won't dwell on this too much. So in our phase 1/2 study, we enrolled boys between 1 to 11 years of age. They must have genetically confirmed Duchenne with mutations in exons 18 and above. They needed to be able to perform a 100-meter walk test without an assistive device, and they needed to have no antibodies to the AAV8 capsid. Now, I know that if you think about some Elevidys, which is commercial, they do have a titer cutoff of 1 to 400. We do not have a titer cutoff. You are either positive or negative. And that's in the initial phases here where we're testing our drug to see the efficacy and the safety. We didn't want to kind of introduce that at this time, but obviously we'll always consider it in the future. We started off with dose level 1, which was the lowest effective dose found in mice. And we started off by giving 1 times 10 to the 14th genome copies per kilogram of our drug. And we gave that to about three patients, and we had a good safety profile. And after that, we had a discussion with an external monitoring committee that said, okay, looks like everything's good to move on to dose level 2. Okay, great. So we doubled the dose. So we then had a dose evaluation phase for 2E for 2 times 10 to the 14th genome copies per kilogram, which is how much viral load you're delivering. We gave that to 2 patients. It looked good. And then we moved on to an expanded cohort to dose 5 additional patients. Then at the same time, we also opened up another cohort because we said, you know what, with newborn screening and everything, like, why not provide access to younger kids who might find out that they have Duchenne earlier on because of newborn screening, or maybe if you have a sibling and you were born with Duchenne and you knew about it, rather than sitting there and kind of waiting for the time when you might be able to enroll in a study, have access to this drug. So we opened it, and actually this dose level 2 in 1 to 3-year-olds is still open at some of our sites, which have not yet rolled over onto the pivotal study. And the reason why they haven't rolled over is because of the bureaucracy of hospitals and like how long it takes to transition. So in the meantime, while they're transitioning to the pivotal version of the protocol, we said, you know what, keep it open. We'll see if we can continue to enroll some patients, and we have. And we still have some slots in there. So basically, you know, we're still dosing some of these kids, and so far everything's been looking really great. So these are 1 to 3-year-old patients who receive 2 times 10 to the 14th genome copies per kilogram. Below, you can see the administration and assessments timeline, which is basically at baseline. What we do is we get a muscle biopsy, we track your muscle strength and function, we get a muscle MRI, and we make sure that you can actually enter into the study based on the criteria with the eligibility. And then what happens is just about in the 2-week period, within that 2-week period before administration, we start to have to discuss starting immune suppression. Some of that immune suppression is weight-based in terms of the timing of when you give it. But typically we start off with, we give eculizumab, we also give sirolimus, and then the day before your dosing, we give prednisone. And this continues at various times, like continues through various times. So basically your eculizumab stops just about 2 weeks after you're dosed. And then you continue with the sirolimus and the prednisone for a full 2 months and then wean off of it. So then by 3 months, you get a follow-up biopsy that will then check your microdystrophin protein expression. So you might say, what's the point of this biopsy? Well, the point is that we want to make sure that not only, you know, that our drug is actually getting to the correct place. So we get the muscle, we wanted our drug to get to the muscle, we get a piece of muscle, we check it, we make sure that we can actually quantify microdystrophin, and we can actually look to see if the microdystrophin localizes to the muscle membrane and kind of rims the muscle membrane there. And you'll see a picture later that kind of speaks to that. That's a process known as immunofluorescence. So we know that it gets there. You can always quantify microdystrophin in the muscle, but you don't know if it's in the right place. But when you look at that immunofluorescence, you could see it's actually going into the sarcolemma or the muscle membrane, and it's getting to the right place. So we get that at 3 months, and then that's it. No more muscle biopsies. And then subsequent to that, we get some follow-up muscle strength and function testing and muscle imaging. And obviously the goal of our phase 1/2 study was safety and tolerability. And here's some of the data that we have for key baseline demographics of the kids that were entered into our study. I'm not going to go through this entire graph, but just so you know, we dosed kids between at screening age of so far 3.7 to 12. I'm sorry, at dosing age of 3.7 to 12.1 years of age. The weight in the 2 times 10 to the 14th genome copy per kilogram, just to kind of show you how much drug we've given because your viral load depends on your weight. We've given up to 35.5 kilograms drug to a child who's as heavy as 35.5 kilograms with, I always say, knock on wood, good safety profile. And the time from dosing that we have right now is up to 19.4 months from in dose level 1 and up to 11.8 months in dose level 2. And our outcome measures that we measure, I'll kind of go through those in the subsequent functional testing slides, but this is just a glimpse of what functional level the kids had at baseline and what we enrolled. We have a little bit more flexibility with what we allow for the time to stand. So in the phase 1 study, it was less than 9 seconds. We didn't have anybody that went beyond 7 seconds. Basically, we didn't have a cutoff for the NSAA and enrollment in our phase 1 study, but that does change a little bit when we move on to our pivotal study. But we had NSAAs in our patients from 14 to 30. And that's the North Star Ambulatory Assessment. And this is a beautiful slide that I hope can stay this way to infinity and beyond, which is our interim safety data. So we have had no serious adverse events in any of the children dosed to date. We have not had any reported adverse events of special interest as it relates to central or peripheral neurotoxicity. This is mainly because of some data that came from other gene therapies not related to Duchenne, and how when you did animal studies, there might be some toxicity to some of the nerves in the spine, and we haven't seen any evidence of that clinically. You wouldn't know, not clinically, unless you sample the tissue, which you would never do unless it was an autopsy related study. So we don't have any data on that, but clinically, no evidence of central or peripheral neurotoxicity. We've had no drug-induced liver injury. We've had no thrombocytopenia. We've had no myocarditis, no myositis. The most common adverse event that was reported with the gene therapy was nausea, vomiting, and fatigue, all of which resolved. And here is some information on the biomarkers that support the consistent, robust expression. So as I told you, the microdystrophin quantification by a method known as Western blot that you can see here on the second row tells us that there is a quantifiable amount of microdystrophin in the muscle tissue. Great. So we're having a great response there. And seeing that, our selected dose for the pivotal study is dose level 2 because we've had a good safety profile. And in our animal studies, we saw that the higher the dose, the better the response. So you can see that in our dose level 2 patients, the average, we only have one patient reported here for their microdystrophin levels in the 4 to 7-year-old age range, but that was 77.2%. And in the 8 to 11 range, that's 39.7% on average, but up to 75.7%. The vector copies per nucleus are how much of the drug can be quantified in the tissue itself. And you can see that listed there, that we're getting a good amount of the drug to the tissue as well. So the drug is getting there. The protein that the drug is supposed to produce is being produced with the microdystrophin percentages. And then the positive fibers on immunofluorescence are how many fibers are staining positive in that rim after you dose with the microdystrophin. And basically, this is change from baseline. And we see that, even at dose level 1, we have, 79.3% positive fibers, 34.6% positive fibers, in the different age groups. And then in the 4 to 7-year-old, we have 71.1% positive fibers. And in the 8 to 11-year-old age group, 45.7% positive fibers. Now, you might say, like, okay, so is this going to get better? Is this going to get worse over time? We don't have a subsequent biopsy to follow up on it, but historically, and what we've learned historically, is that a 3-month sampling of muscle tissue is going to be very early. So like this theoretically should get better over time unless you have an anti-transgene response. But that would manifest as worsening weakness and potentially myositis or myocarditis, which is inflammation of the muscle or inflammation of the heart. So thankfully, we have not seen that again. We're very optimistic based on these results that we will continue to have ongoing good functional benefit from the use of RGX-202. So the natural history that we're using in our study in the subsequent slides to review functional data with you is something that we got from external data sets that are available to us. So not just to us, they're available to everybody anywhere. Obviously, you have to purchase that data, and then basically we can match our patients' baseline phenotype or descriptors based on the North Star, a child's base. Like, say, for example, we have an 8-year-old patient. 8-year-old, what's their baseline NSAA? What's their baseline time to stand? What's their baseline 10-meter walk run? What's their baseline time to climb, for example? And then we can try to match them for all of these at baseline. And then we compare to what we see the external control do at 12 months or 9 months, for example, which I'm going to show you shortly. So we have 12 months of data for our dose level 1 patient and 9 months of data for our dose level 2 patient. So we don't look at where they are later, but like where they were at the start and how the other children that we're comparing to performed at 9 months later who had the same baseline picture, or how those with the same baseline picture performed 12 months later. And then we compare and see if there's any improvement as compared to an external control. But we also will show you some change from baseline. So our dose level 1 participants demonstrate improvement in function and exceeded external controls at 12 months. So just to kind of walk you through this slide here, the axis to the left here shows a decrease in time, which is technically an improvement, right? Because you're doing it faster. So you can see in blue the change from baseline at 12 months, but you can see in gray the predicted change that you would expect for someone who's matched to the kids in our study with an external control. And basically, compared to an external control, we had an improvement in 2.9 seconds in the time to stand, an improvement of 1.1 seconds in the 10-meter walk run, and an improvement of 1.8 seconds in the time to climb. So as you know, the predicted trajectory of a child with Duchenne is typically decline. So we are not only not declining, but also improving. So that's where we're getting these changes. And then for the North Star Ambulatory Assessment, you can see that as compared to an external control, we had an improvement of 4 points on the score of our North Star. Now, in order to get a drug approved, you have to go through discussions with the FDA and present your data. And obviously, we have the benefit of looking at what Elevidys went through with the FDA. And one of the things that the FDA referred to as something that helped with their approval is something called a minimally clinically important difference. And this is based on natural history data that was calculated in a study written by a group that also included Tina Duong. And basically, they calculated velocity at 12 months that would indicate a minimally clinically important difference. So that minimally clinically important difference is designated in the gray boxes here. And you can see that our kids at dose level 1, which is not the dose that we're taking to pivotal because we're trying to go for even better, at dose level 1, our kids performed better than that on time to stand velocity, 10-meter walk run velocity, and time to climb. This does not exist for NSA, the North Star, so we couldn't put it up here. And we don't have MCID data for 9 months. It's only listed as 12 months, so we can only compare our dose level 1 patients to that. Now, in our dose level 2 patients, we also saw tremendous improvement. So again, looking at how you would expect our patients to perform. So this is at 9 months, not 12 months. So it's a little bit of a sooner time cut that we took. But at 9 months, and hopefully anticipating an even better response at 12 months, at 9 months, we saw in our 8-year-old patient, so these are older patients, as you know, the older the patient, going back to the slide we're kind of speaking to, which I didn't really delve on here, the NSAA, but a lot of the function of the patient kind of follows this trajectory where kiddos might have slowed development initially, but usually get picked up with their diagnosis around 4 or 5. They may gain some function, but then start to peter out with their function after the age of, like, 6.5 or so. You get a rapid decline phase. So, in spite of our kids being in a rapid decline phase, we do have improvement in the time to stand at 9 months of 0.9 seconds. Our 10-meter walk run improved by 0.7 seconds, and our time to climb improved by 1.2 seconds. And in our 12-year-old patient, he improved at 9 months by 3.1 seconds in the time to stand, 1.3 seconds in the 10-meter walk run, and 2.7 seconds in the time to climb. And in the NSAA, the North Star, we found a match at 9 months for the 8-year-old, but not for the 12-year-old. Hopefully, we'll get a match at the 12-month mark for him. But there was a improvement of compared to natural history of 2.8 points on the NSAA. So again, just kind of hammering home that these are older patients, not something we've seen with some of the other studies that have been reported in the younger kids, like between 4 and 6. This is an 8-year-old and a 12-year-old improving with our drug. And this is just an average of the two together. So overall, the data is an improvement of 2 seconds in the time to stand, an improvement of 1 second in the 10-meter walk run, an improvement of 2 seconds in the time to climb 4 stairs, and an improvement of 7.3 seconds in the North Star. And obviously, these data will become more robust as we get more patients in. So we also did caregiver reports, and the caregivers reported improvements in the home and in community environments as measured by the PODC. And the improved skills that they included were running, riding a bicycle or tricycle, climbing stairs, walking in the community, and participating in recreational activities with sports and peers. So overall, we've had positive safety, biomarker, and functional data that demonstrate the potential of our RGX-202 drug, and it's been well tolerated in 11 patients across both dose levels with no serious adverse events or adverse events of special interest. Our biomarkers support consistent, robust expression and transduction, which means that our drug is getting to the right place, it's actually producing protein, and it's localizing to the muscle membrane. And our participants with RGX-202 demonstrated clinically meaningful improvement in functional outcomes at both dose levels, exceeding comparisons to natural history controls and exceeding the minimally clinically important difference. Now, that's our overview of the phase 1/2 study. I'm going to tell you about our phase 3 study, which is just a study overview. We don't have any data on these patients, but after we had that data, we had to get some alignment with the FDA on the pivotal design. And our goal is to use RGX-202 microdystrophin as a surrogate endpoint to likely predict clinical benefit, hoping that they will accept that our microdystrophin levels that we can quantify in the muscle tissue will predict clinical benefit. And we expect to file a biologics license application in 2026 using this accelerated approval to include approximately 30 patients at dose level 2. And in the pivotal study, we are now enrolling boys ages 1 and above, weighing 10 kilos or more. The reason why we put 10 kilograms is because we have to do safety labs, and doing safety labs on anybody less than 10 kilograms is really restrictive. So we had to start at 10 kilograms. The children also need to be ambulatory at this time, so they just need to be able to walk 10 meters on their own independently. They should also have genetically confirmed Duchenne mutations in exons 18 and above, but stay tuned because that is changing soon, very soon, the beginning of this year. And they should also have no pre-existing antibodies to the gene therapy, which would be the AAV8 capsid. And our primary endpoint for our pivotal study will be the proportion of patients with greater than or equal to 10% of microdystrophin levels. So we are anticipating that all our patients are going to produce at least 10% of microdystrophin levels across the board and across all patients, like meaning everybody's going to have some microdystrophin. Secondarily, we're going to be checking for functional tests. These include the timed function tests like the time to stand, the time to run, walk, and the time to climb. And in our younger kids, we are also going to be using the SV95C, the stride velocity 95th centile, which is a wearable because these kiddos won't be able to participate in these timed function tests as well or do the NSAA as well. And then we have some exploratory endpoints, and this is just a high-level overview of our study, but the exploratory endpoints also include the North Star Ambulatory Assessment and the SV95C for the 4 and over-year-olds. We have multiple sites open across the country, and we will be able to update this very shortly as well, as Pivotal is up and running in most places now. Right now, we have a site in Virginia Commonwealth University, Lurie Children's, sorry, Arkansas Children's Hospital, University of Texas Southwestern Medical Center, and Stanford. All right, and just a little plug here for our Affinity Beyond study. So this is an anti-AAV8 antibody assessment study. This is a study where it's non-interventional, but basically we want to understand the prevalence of AAV8 antibodies in boys with Duchenne and identify any kid that might be able to participate in our study and kind of get fast-tracked into our study. And basically, this is set through the hub of rare disease research. The PI there, Dr. Han Fan, will have a video visit with you, and a nurse will set up to come to your house to do the blood work. That will then be sent out. And we are set to enroll up to 200 patients. Right now, the key eligibility is boys ages 0 to 11, and that's going to expand soon, given that our pivotal is now 1 and older. And they also must have a diagnosis of Duchenne, but no mutation exclusions. And of course, they must have not received any prior gene therapy product, because that can alter the results as well. And I just wanted to acknowledge everybody that's been participating. Obviously, all of your family, the most important are the families that have put their trust in us as a company to participate in our study. We're doing our best by you. And obviously then our investigators at the individual sites also working very closely with us to provide this care to the families and make sure that it's given safely. Very, very important are all the study coordinators who do all the operational work to make things happen. And obviously our wonderful team at REGENXBIO, who always has the best interest of your kids in mind and is always here for you. And of course, to the Jett Foundation, thank you so much for giving us a place to kind of talk about our study. And then I guess we'll move on to questions.
Perfect. Thank you, Naz. That was
Yeah.
wonderful. Really appreciate you taking the time and also breaking it down too into simple terms. So really appreciate that. If you guys do have any questions, please again put them in the Q&A box. I have 2 to start us off. Will the RGX-202 trial be expanded to the UK and the EU at any point? Are there plans to do so?
Yep, we are definitely seeking to go to Europe, but our first place that we're aiming for at this time is Canada. And then it's just like one step at a time with all the regulatory work to get the drug in the different places.
Absolutely. And then
We are open to people from outside the US reaching out to be dosed inside the US. And we are in the process of hopefully soon, I would say, don't quote me on this, but maybe by April, having a mechanism in place to get you tested for antibodies through the Affinity Beyond study outside the US with the same mechanism.
how long would someone have to stay in the United States after being dosed?
So after being dosed, there are very frequent visits for safety monitoring that happen. I would say at least 3 months, um, with the hopes that you would come back monthly until month 6. And then it kind of tapers off to like every 3 months, and then it would be 6 months. We do have a long-term follow-up. That's something that's required of us by the FDA. So you have to have long-term follow-up for up to 5 years after dosing. So that would go on every 6 months for 5 years. But
Thank
it's
you.
good because we can make sure you're doing okay, and obviously the travel costs would be covered too.
Awesome. That's good to know. I think that's just something important for people to know when considering that, right?
Yeah.
We do have a question, so I'm gonna kind of all encompass this one, but combination therapies. So can the child be on drugs like Duvizat and some of the others that have been approved in this space?
Yep. Great question. So Vamorolone, we have allowed for inclusion of vamorolone in our study, in our pivotal study. But that has a transition period because we're not totally clear on how immunosuppressive vamorolone is compared to prednisone. So during that time when we have the immune suppression regimen, we do ask that the families transition to prednisolone just for that like 3 month period, like 2 month full dose and then weaning off. But they can stop it like, just the day before the dosing when they start the prednisone, and then they can restart it right after. And we'll get the baseline functional testing on the vamorolone, just transition for 3 months on the prednisolone, and then basically we'll get the follow-up functional testing like later on vamorolone again.
Okay, perfect.
Obviously it kind of um going back and forth muddies the water, but we're hoping that because we're trying to meet the need of the community, because some folks will be on vamorolone now, but we're hoping that the transitional additional like the transition to prednisolone, that effect will wear off at least like 6 months after or 3 months after.
Perfect. And then what about,
Oh,
uh,
Ruby's at? Okay.
and also, just as an encompassing other exon skipping therapies that
Yep.
someone may have taken already?
So that's something we're very interested in as well, like trying to address the need in the community. The issue with Dubizat is that it has the side effect of low platelets, and so does gene therapy potentially as a risk. So we definitely don't want to introduce that when there's a high risk of thrombocytopenia with gene therapy. But we are working on ways to potentially incorporate that. The part where it gets confusing is that if you look at the readout for Javinostat, what they got their approval on was like 18 months after therapy. So 18 months after starting Javinostat, you may see an improvement in function, and if we're doing our readout at 24 months, how do we distinguish what's improvement from our drug versus what's improvement from Dubizat? So it would probably have to be like a separate trial for combo. But that's something that we've considered, but obviously it's still kind of in the research books.
Yeah.
For exon skipping, it's the same thing. Particularly right now when we're looking to look at immunofluorescence in particular to see how the dystrophin is localizing. Like even if we were to stop the exon skipping therapy, we don't know how long it would take for that to wash out. And then like would we just keep it on board the entire time? What is the safety of keeping it on board? We're obviously thinking about it, but it's still a work in progress.
Yeah, absolutely. Thank you for the transparency. I think that's really important. Can someone who expresses non-functioning dystrophin participate?
So we do not require a baseline biopsy to be in our study. So we won't know if you have non-functioning dystrophin until you get your baseline dystrophin. And your baseline biopsy, and then it takes like weeks for it to come back. So what we just require is that you have a clinical diagnosis of Duchenne. So if the primary investigator finds that your mutation, even if it's in frame technically, because there are some parts in frame, because I know some studies exclude in frame, but in frame in the actin binding domain might be more of a Duchenne phenotype. But if even if you have a mutation that's questionable of whether or not you might have a Duchenne or a Becker phenotype, if you're old enough where you're demonstrating that you do have a Duchenne phenotype and the primary investigator thinks that your clinical picture is consistent with Duchenne, we will dose you.
And then I know you said right now, you have to be ambulatory. Are there considerations being made for non-ambulatory cohorts possibly in the future?
Yep, in that same discussion with Javinostat and antisense oligonucleotide testing
Perfect.
administration. Yep.
Um, Fair question. It's my understanding that many patients on other gene therapies lost benefits from it after a few years. Is there anything in REGENXBIO's therapy that would suggest a different outcome slash longer lasting effects?
Yeah, so we have a couple things that are all just speculation at this point, right? Because we haven't dosed anybody for up to 3 years or 4 years or 5 years yet. So the trial is trying out our drug and seeing if it actually works and if it's sustained. Um, we do think, like, again, based on the animal studies, that our C-terminal domain will help out. And honestly, our immune suppression regimen, if you are, you can start forming antibodies to the drug within hours, maybe even less than an hour of administration. So if you're actually suppressing the immune response to the drug, are you allowing for better entry of the drug into the muscle tissue? It's possible. There's not going to be a way for us to distinguish that because we're not doing a study with our immune suppression regimen, but without. But do we hope that that's actually helping with what we call transduction? Yeah, for sure. And also durability of the drug, for sure. So we hope that these things together, and obviously we use AAV8, is that a more tolerogenic vector to use, like where your body doesn't kind of fight it off, like maybe some of the other vectors and whatnot.
Yeah,
So
absolutely. Um, I think you already answered that. And then overseas, if anybody does have any questions about overseas specifically, I would reach out to the REGENXBIO team. You can reach out to us and we will go ahead and send it over to them. Um, and then if anybody has any other questions, I'm just going to look at Facebook to see if any came through on there. Nothing on there. Um, Again, Naz, thank you so much.
yeah,
If
sure.
any other questions that we didn't get to, or we all know it takes some time to process and think of
Yeah,
questions sometimes,
yeah,
please feel free to send them our way and we will send them over to the Gen X Bio team and try to get some answers for you. And then again, if you're watching the recording, please also do the same. And
yeah.
with that, I will say thank you again to everybody who was here to learn today. Thank you, Naz. This was wonderful.
It's a pleasure.
We appreciate all of you. We appreciate REGENXBIO Bio for making this possible, and we will see you all again soon.
Thanks.
Have a good
Thank
day.
you for having us.
Bye.
Bye.